Hello, my name is Esmeralda Langarica, and I will be focusing on talking about the unraveling of molecular landscapes of lung adenocarcinomas, insights from integrative omics analysis. So, the research article that I'm going to be focusing on delves into the intricate role of DCAF in the context of human lung adenocarcinomas, LUADCs. Specifically, it investigates how DCAFs contribute to the molecular landscape of LUADCs. So it can unravel key biomarkers that can help in disease diagnosis, prognosis, and targeted therapies. So the key objectives of the study is understanding DCAFs in LUADC development, and to understand the functional significance of DCAFs, such as DTL, DCAF4, 12, and 13, in driving the development and progression of LUADCs. Secondly, identification of prognostic biomarkers. So it's important to identify the potential prognostic biomarkers among DCAF that could help stratify patients based on disease severity and guide their treatment decisions. Lastly, exploring therapeutic targets. The study explores DCAF potential therapeutic targets, hoping to uncover different targeted therapies that can improve patient outcomes and survival rates. So, a methods overview. The study used an integrative omics approach to comprehensively investigate the role of DCAF in LUADCs. These proteins that associate with DACF, a ligase complex. This complex is involved in subsequent degradation of target proteins within cells, regulating various cell processes. Dataset selection. Multiple datasets were made and utilized for this analysis. You can see 3 of the GES. I'll get into those a bit later. These datasets contain valuable gene transcript data from both LUADC samples and normal lung tissue, providing a comparative basis for the study. DCAF expression analysis. The study focuses on 19 well-defined DCAF and investigates their expression levels in LUADC compared to normal lung tissues. Analytical tools that were used were GO2R, which allowed for a comparison of gene expression patterns between different sample groups. This analysis helps identify DCAFs that are significantly upregulated or downregulated, and LUADCs. Genomic data analysis were also performed using cBioportal, and statistical validation was used to make sure to ensure the reliability of findings, to assess significant differences in gene expression levels and DNA copy number alterations in LUADC samples. So, as mentioned before, they did a comprehensive search for DCAF genes using Unigene and GeneCard databases. So they searched it, and it resulted in 19 DCAFs that were used in their study, which is the mutation frequency of DCAFs in lung adenocarcinomas. So as mentioned before, gene transcription data of normal and tumor tissues were obtained from the NCBI and the gene expression, so GEO. It was obtained for 3 genes, so here we can see GSE19188, GSE198004, and then here next to it we can see that it says genome-wide screening of transcriptional modulation in non-smoking females in lung cancer in Taiwan. So once you pick and define your groups, I wasn't able to replicate exactly because I don't know exactly how they define their groups. It doesn't really say in like the study, but once they define their groups and they filtered it how they wanted it to, they would look for the genes that I previously mentioned to see if there was an upregulation in it. So here we can see what I highlighted, like the defined groups that they can put and they can choose. All these different columns have different categories, so they could choose and see how they wanted to filter it. So for the first part of results, they saw elevated mRNA levels in DTL, DCAF 4, 12, and 13 in LU-ADC. They also saw reduced mRNA levels in VPR, VP in LU-ADC. Here from their results, we can see a correlation between transcriptional factors of DCAFs and gene copy number variation. We can see frequency of gains and losses among specific DCAFs in LU-ADCs. And then finally, we can see there's an association between DTL expression and overall survival of LU-ADC patients. This survival analysis results in significance of DTL as a biomarker. So from these results, they concluded distinct impacts of DCAFs in LU-ADC development. They saw specifically in DTL, DCAF 4, 12, and 13, they showed consistent elevation in mRNA levels in LU-ADCs compared to normal lung tissues. These findings show the heterogeneous nature of DCAF's involvement in LU-ADC development. This suggests varied roles and potential biomarker significance. Secondly, potential roles of DTL, DCAF 4, 12, and 13 as oncogenes. They saw a consistent elevation in DTL in LU-ADCs and a negative correlation with overall survival. They also saw DCAF 4, 12, and 13 showed elevated expression patterns, suggesting their potential roles as oncogenes in LU-ADCs. Implications for prognosis and therapeutic strategies. So the identification of these as potential oncogenes in LUADC hold a significant prognostic implications. These show the potential for prognostic biomarkers. Um, Further research into these DACFs, their interactions with other genes, and their impact on tumor progression is crucial for developing targeted therapies and improving patient outcomes. So the utilization of multiple datasets provides a comprehensive analysis and strengthens the generalizability of findings. Limitations. There's a potential variability and biases in the datasets that could impact the accuracy and reproducibility of results. Assessment of identified biomarkers. So the relevance and significance is that the elevated expression of these biomarkers, along with VPRBP, shows the potential biomarkers with prognostic implications in LUADC, which can serve as indicators of disease severity, progression, or response to treatment. Discussion for biases. The only bias is the variability in sample characteristics. And then implications for future research and clinical applications. Further validation studies are needed, like experimental assays, validation of cohorts, are important to further identify these biomarkers, and clinical applications, integration of validated biomarkers into clinical practice may enhance LUADC prognosis prediction, treatment, and monitoring of treatment responses. The incorporation of DCAF based biomarkers into precision medicine approaches can help personalize lung cancer management strategies. Thank you so much for listening, for your time. Looking forward to your to any questions, comments, or concerns. Thank you.